அனைவருக்கும் வணக்கம். Very good evening to everyone. Good morning, Dr. Sarthak, Professor Pandian, and Dr. Radha Pandian, if she's here, I'm not sure. And I would like to welcome you all to today's webinar by International Fertility Research Foundation on non-invasive pre-implantation genetic testing, developments, concerns, and opportunities. Today we have with us Dr. Sarthak Savarkar from New Jersey, and our chairpersons for today are Dr. Devan Patel and Ms. Sarguna Devi. So Dr. Devan Patel is a consultant embryologist and scientific advisor at Nova IVI, Ahmedabad. Sir has done his master's in veterinary science from Indian Veterinary Research Institute. Sir has also done his master's degree in biotechnology of human-assisted reproduction and embryology from IVI Foundation, Valencia, Spain. Sir has been working in human ART since 2007 and in bovine ART since 2002. Sir has previously worked as regional manager in embryology with Nova IVF Fertility. Sir has worked for more than 11 years with Dr. Manish Bankar, the leading infertility specialist in India. Sir has been awarded Hall of Fame for the embryologist for West Zone by ET Healthcare Awards 2019. We welcome you, sir.
Thank you.
We have with us Ms. Sarguna Devi Sivanandam. She is a senior clinical embryologist with more than 15 years of experience in the field of ART with a HPC UK registration. Ma'am works as the lab director at ARC Hospitals and Research Center, Chennai. Ma'am has earlier worked at Professor Simon Fisher's Care Fertility, Northampton, UK, the Sims Clinic, Dublin, Republic of Ireland, Kraft Hospital and Research Center, Kodungalur, at Women's Center, Chennai, Manipal and Anpur Fertility Centers, Bangalore, and Prashant Fertility and Research Center, Chennai. Ma'am has published various research papers in national and international conferences. Her interests are low-cost IVF, advanced sperm selection, and non-invasive embryo selection for repeat implantation failures. We welcome you, ma'am. I now request Dr. Devan Patel to introduce our speaker. Over to you, Dr. Devan Patel.
Thank you, Dr. Pavitra, for a nice introduction. And before introducing Dr. Sarthak, I would thank Dr. Kritika Devi for inviting me as a chairperson, co-chairperson for this session. And I think this is currently one of the very latest topic which I would like to really understand as an embryologist also regarding the non-invasive PGTA. So today the topic will be presented by Dr. Sarthak Savarkar, who is the founder and chief executive officer of Rosa Biotech, Princeton, New Jersey. Previously, he has worked as chief operating officer at MedAnswers, California, and he has previously worked in Cooper Genomics in various positions, starting from research associate to research coordinator, and finally as a director research. He also has research projects, some poster and oral presentations at international conferences like ASHRAE and ASRM. The prominent publications are Reproductomics, publication in 2018 as a chapter on reproductive genetics titled The Genetic Selection of Human Embryos from Fish to NGS, Past and Future. He is also having a chapter titled Current and Novel Methods of Chromosome Testing in a book in vitro fertilization, which is a textbook of current and emerging methods and devices. I would request Dr. Savarkar to take over.
Thank you, Dr. Patel. I would, firstly again like to take this opportunity to thank honorary chairpersons Dr. Patel and Dr. Devi, Dr. Sarguna Devi, to having invited me here. Of course, Dr. Pandian and President Dr. Kritika Devi also, and other esteemed committee members. Just to begin with, today is going to be a very non-commercial and sort of an academic presentation here that we've set out. Rosa Scientific is a biotech company that we founded with a singular goal of creating novel in vitro diagnostics. And we want to do this in a highly replicable fashion. But at the same time, we are also focusing on improving existing diagnostics in the market. And for doing this, we are using some, we have an amazing team, highly qualified team from all the premier institutes Berkeley. I have team members from MIT who are using their expertise, their cross-field expertise in biochemistry and artificial intelligence to improve in vitro diagnostics. And for this particular project, this particular project is a part of Rosa Fertility, which is a concern that is operated under the fold of Rosa Scientific. What we saw during this research is that we have several benefits, concerns, and opportunities that we came across for NIPGTA. It's a mouthful, so I'm probably going to just call it non-invasive technique from here on out. And that's what we will chat about today. One quick rider is that this talk might be slightly more technical, but it is intentionally left so. One of the goals for me today is that I want experts like you to challenge and demand more technical detail from scientists like me when we present data to you. Let me now start sharing my screen here real quick. 1 second. Are you able to see this?
Yes, yes. Kindly change to presentation mode.
So without further ado, I'll quickly jump into what the agenda that you set out for today is you all are very well aware of PGTA, what PGTA is, what it looks like today. So I'm going to breeze past that, initial introduction slide, but I would be remiss if I don't mention it. Then, on the background of that, we will explore what the non-invasive assay is, how it works, the clinical efficacy around it, and then obviously the benefits around that, and more importantly, we'll talk about the concerns towards the end of the presentation. And obviously through the concerns, what we will identify as a few opportunities for us to improve the assay with. Quite honestly, I'm a trained biochemist, and I'm learning a lot with my team when I'm looking at the AI models and the technical details that surround us actually going in and looking at more detail in terms of how the improvements could be. So even if we try to just see as to how and where the field is headed, I think this would be beneficial for everyone. Please feel free to interrupt me whenever you feel like, or we can always have the question answer session at the end of the talk. So you've seen, this slide presented to you in various flavors. Why use PGTA? We have seen increased implantation rates. We've seen a reduction in miscarriage rates, faster time to pregnancy, and more importantly, what we've seen PGTA do is enable a confident single embryo transfer compared to multiple embryo transfers that were predominantly used in the field earlier. And when you use PGTA, in the traditional sense, we use embryo biopsy. So let's quickly take a look. There are some pros for embryo biopsy. As I said, there's improved outcomes. Rather than looking at an embryo in strictly a morphological sense, we have a better qualitative and quantitative predictive assessment when we have when we started using cells that we derived from the embryo to make genetic predictions. There's been a rapid field-wide adoption for this. We're not going to use today's presentation at all in terms of whether you think that PGTA is right or wrong. But wherever people have used it, it's been uniformly accepted and adopted in a very organized fashion. So because of that, we've been able to generate large amounts of data, and that has been indicative of genetic health of embryos, and we've improved our understanding of what an embryo looks like using PGTA. And that's why embryo biopsy has been actually pretty good for the field. When we have an embryo biopsy done, we have basically cells, and cells have a lot more information, which basically can be used for different outcomes like PGT for monogenic diseases, structural rearrangements, as well as aneuploidy. So there's a lot more comprehensive genetic information that is available. But then there are some cons of the embryo biopsy, and that's why we want to find out a non-invasive method of doing the same assay. So what are the cons here? So embryo damage, and this is pretty self-explanatory. If you want to disturb a fragile system like an embryo by actually going in and biopsying it, there is some risk of damage. We've mitigated that risk by actually working with blastocysts rather than blastomeres in most cases. But nonetheless, it's a non-zero risk of damage to the embryo. And the other aspect that is very important to look at is that this is very dependent on the skill of the operator. And it's not possible to have that uniformity across the board and across the globe. We'll explain. We'll explore the reduced sensitivity and specificity part in a slide where we'll see as to what is the origin of embryo mosaicism very quickly. But this is again a con of embryo biopsy, because sampling is biased as to you're looking at a snapshot within the entire picture. So there's a good chance of embryo mosaicism that can result as a part of embryo biopsy. The last point is patient resistance is more of a cultural con, a cultural limitation here, but patients are hesitant when you say that you're going to take a piece of the embryo out, and that has been always slow for adoption of PGTA. All of these points are something that we all know of, and then there's one more aspect of this where this is a publication by Dr. Mune, where they looked at the number of biopsy T cells and how it affected pregnancy outcome. And what they saw was really interesting. They saw that if you have less than 10 cells, you have a higher ongoing pregnancy rate, whereas if your biopsy was greater than 10 cells, then you have a lower ongoing pregnancy rate. And this is statistically significant for us. So this variation, coupled with the fact as to as I was talking about earlier, the bias of sampling, the chances of misdiagnosis are higher just by the virtue of where you are going to sample. If you have some euploid cells being sampled along with aneuploid cells, which are a result of mitosis, there's a good chance that you're going to start seeing mosaics, and again, increased chances of misdiagnosis. So do we have an alternative for biopsying the embryo? And that's what we started seeing, is that people started testing blastocele fluid, and they were aspirating blastocele fluid and testing it. They found that it did actually contain genomic DNA in it. So then a latter question that was obviously asked almost immediately was, would spent culture media have genomic DNA in it? And we started seeing evidence that even spent media had genomic DNA in it. So that basically brings us to the non-invasive PGTA assay. Before we delve into more details about what the assay is, let's look at the pros and cons of the non-invasive assay. With the name comes the first pro, which is it is non-invasive. Again, you're not touching the embryo per se, and you're not following any active biopsying around it. So the chances that you would hurt the embryo are minimal. this results in a cost saving for biopsying the embryo. And more importantly, I think the last point is something which is very interesting for a diagnostic company like us, because the technical challenges of implementing something like this are extremely low. We can have relatively low trained. I'm not going to say that we can have untrained people doing this. We still would need embryologists working in the lab, but at the same time it reduces the challenge, because you're simply aspirating out a few microliters of media and packing it and sending it to the lab for testing. This can basically uniform collection practices will be a lot easier to implement when we use non-invasive assay. It has some cons too. Maternal contamination, especially if there's some cumulus cells that are hanging out by the embryo, there's a significant chance that there could be maternal contamination. There'll be, we ourselves are working with some innovative techniques. We are working on patenting some really innovative techniques to come around maternal contamination. But nonetheless, it's one added step, and we absolutely have to be certain that we aren't seeing that. Then the second aspect of this is, the origin of the cell-free DNA that's present in the media. We don't know where this DNA is coming from. We don't know if, what is the nature of fragmentation? Is it sticky-ended DNA? Is it blunt-ended DNA? We are not sure. So that is something which we have to actually, spend time researching. There's some interesting research by Dr. Svetlana Majdukova's lab that I've seen, but there needs to be a lot more research as to find out where this stems from. We'll explore this point a little bit more in detail in a few slides. Lastly, why the origin of the cell-free DNA is important is because the assay that we develop around this has to be sensitive enough to the nature of fragmentation, to the origin, and that is something that we are still struggling with as a field. We're still trying to work around this as a field. So just quickly taking a look at how it works, we have the non-invasive chromosome screening technique. So you have oocytes, basically you fertilize them, usually culture them to day 5, day 6. I say this, there are groups which are culturing, in continuous media, they're in sequential media. There's different kinds of culture practices that are being used, but our assay that we are working on is flexible around day 5, day 6, collected media from both sequential as well as continuous media. This is collected, then there's sample preparation, there's next generation sequencing, and then you have your result. So the amplification aspect, the amplification assay that I was mentioning for, there's multiple variants of this. Sureplex, MDA, Malbec are commonly used. Malbec, MDA being sort of the favorites in the field. Conventional, amplification methods like Sureplex are nonlinear. What that means is that every time any part of the DNA is amplified, the amplicon is used as a template for the next version, the next cycle of the amplification. With Malbec and MDA, being quasi-linear, they basically prevent the original amplicon from being used as template, and that basically results in improved sensitivity and specificity for PGTA. Is this a desired outcome? Is a question that Rosa is asking, and we will see that in the next in the presentation. And this is the summary of the results that I said that I would present for all the non-invasive assay groups that are there. There might be 1 or 2 missing that I might not have on this slide, but this pretty much captures what we want to show for the summary of the results that are across that are observed across the groups. You have different you have different variables here. You have fresh media collection versus from frozen embryos. You have blastocyst fluid, and sometimes you have spent media, but you have spent media and blastocyst fluid. Then they're looking at actually the rate of amplification, and then you're looking at ploidy concordance. We tabulated these to sort of just look at them a little more pictorially, and they're just easier to understand. But what you'll see is that you have essentially, ECM, blastocyst fluid has a very low concordance. But if you look at ECM, you sort of start having, they're all across the board, but you start seeing a little bit more with ECM. You have a better chance with frozen embryos. And if you have ECM plus blastocyst fluid, groups have reported 100% accuracy for ploidy concordance, sensitivity and specificity. So, I mean, that's it, right? We've achieved 100%, so we don't need to do much more beyond this. But no, it doesn't work like that ever. So let's start looking at the concerns that we have with current NIPGTA assays. And that's where we sort of come to, the origin of the DNA, as I had earlier mentioned. Cumulus cells could be a potential contaminant. So, and that is one of the one reason why there could be DNA in the media. The primary hypothesis in the field is that apoptosis, or programmed cell death, is the primary contributor of cell-free DNA. And sorry, excuse me. Apoptosis basically results in non-random degradation, and the fragment sizes are 180 to 200 base pairs. And the other hypothesis is that necrosis could be the resultant of the DNA. And in this case, what you start seeing is that this is randomly degraded DNA, and the fragment sizes are somewhere around 500 base pairs. It could also be secreted DNA that's coming in from the inner cell mass, or it could be extrachromosomal micro DNA. Why this is important is because where the origination of this DNA is going to actually tell us as to what is the true representation of the genetic fate of the embryo. So if it's secreted DNA coming from inner cell mass, it's probably going to be the fate of the embryo from this assay. The detection of aneuploidy is going to be more representative of the fate of the embryo than, for example, if it is coming from extrachromosomal microDNA or cumulus cells. So that is why I think we need to understand more in detail about the origin of DNA. I've been working with University of Kent and trying to, and we are trying to work with this problem. But there's some other problems. These are more esoteric problems, right? I mean, where the origin of DNA is something which obviously is an interesting problem. But I think there are some real case. There's some real issues with the assay, and for this particular one, we're going to look at the amplification system of Malbec. Malbec, as I said, was quasi-linear. There are some superior parts associated with Malbec. It uses completely random primers. What this means is that the amplification is going to be all across the board. There's traditional amplification bias, with just known primers is going to be obviated by this. Then we are not using the amplicon that is generated, and that also results in reducing of PCR bias. We have seen that Malbec is very good at amplifying GC-rich regions, and there's uniform genome coverage up to 90%. And this essentially leads to a lower allele dropout compared to even other assays like multiple displacement amplification, MDA. the problems that Malbec has reported is that the polymerase is relatively error prone. So monogenic diseases as a part of testing with Malbec is a limitation at this point in time. It's a temperature sensitive protocol. So variations across performance in different labs have been seen. Could there be there is a GC bias, and that is the other end of the fact that it can amplify GC rich regions. Genome coverage has been shown to be about 90%, but significant chunks of the genome are consistently underrepresented. So you might have seen this in the lab, or you might have seen this at presentations, but what are we looking at here? Are we looking at a noisy euploid, low-level mosaic, or is it just telomeric loss? How would we classify this? One of the problems that we saw is that lab folk usually end up looking at data as gospel, that the machine is just spitting out gospel. Nobody's challenging what they're looking at, especially when it's an important indicator like telomere shortening, which could potentially be a very significant indicator of fetus elimination. So if this were noise and Malbec has uniform coverage, then why do our profiles look like this? And there could be several reasons for this, right? I mean, it could be a true biological representation, unlikely, but it still could be. There could be consistent genomic underrepresentation, or the slightly unthinkable in terms of a highly vetted assay like that is that could there be some kind of amplification bias? So amplification bias, has been reported because Malbec, as we said, has GC, bias included in it, and GC bias is common across whole genome amplification techniques. But this bias could potentially, be underrepresenting some yields, and this could be actually, falsely, it could be a false positive for allele dropouts. But isn't this like the pipeline job to catch it anyway? Yes, when you look at Malbec's report of 90% of coverage, what are we using for this, right? We are using old statistical techniques. And what that means is that we are using a very, very highly variable bin size. I was trying to think about how to explain, bin size for this talk, and I quickly think of bin size as something like a pixel, right? So lower resolution will basically end up yielding a higher false positive for allele dropouts, because if you're looking at just bigger picture, you might not see what's going on the smaller. If you had smaller pixels, you probably would have seen something which is missing. But on the contrary, what happens is that if you have a higher resolution, it just becomes all of it just becomes noise, and this could also confound classification. So what's going on? And that's what is a question here. Is the problem is that bin variability is too large, and that is actually preventing us from distinguishing between underrepresented regions. Amplification bias is something that that is a result of that. We have to start reimagining things. We are using statistical techniques and analysis softwares, which at its core was math written about a decade ago. We have to start reimagining, and we have to start using newer statistical techniques here. I know I'm running a little bit behind, so I'm going to quickly go through. But if you look at this, Malbec, for example, there's about a 10%. If the resolution is set at 750 kilobase pairs, we see that 90% coverage. But allele dropout with reducing and increasing of with increasing and reducing of the resolution is something of a very it's a true problem here. So high variability in read distribution is one of the cons that we can add on to the Malbec problem. This is interesting. We had about 5,315 embryos that were evaluated using this particular NIPT assay at New Hope Fertility. The unpatterned gains and losses across this heat map are expected, right? They're true. But then there's something, for example, chromosome 13 has a uniform loss across the board in all patients. Now that's a troubling trend of losses. And this is, probably a false positive for allele dropout. So unexplained amplification bias is another aspect of Malbec in terms of cons that can be added. So can we do something? Yes, and that's exactly what we want to do is we want to create true reference pools, and use machine learning to actually train the algorithm to read between the signals that are coming from the machine. And the instrument. So ultimately, what really matters is, the last of this is basically the proof in the pudding, as they say, is basically if we are able to actually correlate our assay directly with ongoing pregnancy rate, fetal heartbeat, then you obviate the need of finding ploidy concordances with trophectoderm or whole blastocyst. And that's something that we are working on. We're working on as we speak. Our bioinformatics team and our AI team is essentially working on creating algorithms which will distinguish signals a lot more. We are working with true instrument data rather than working on some previously normalized data there. So, and by developing the next generation of the non-invasive assay, we should be able to utilize our synergies between chemistry and bioinformatics and create assays that are more determinative of outcome. I'd like to quickly thank the Rosa team, Dr. Mune, Josh Sams, Dr. Verma, Ms. Haran, Dr. Christensen, Alex, and Danny Barak. These are all superb scientists who have helped us create some really cool techniques and some data that we'll be presenting to you over the next few months. Our collaborators are Embryo Genetics, New Hope Fertility, University of Kent, and the Zuis Foundation for Reproductive Medicine. And lastly, this is my 21-day-old baby that I would like to thank because she let me actually prepare for the talk, and she actually let me sleep for at least a few hours so that I'm ready and not a zombie in front of you. Thank you very much.
Thank you for a nice talk.
You're welcome. I was trying to walk a line between making it too technical, not just because of the fact that the audience here, but even my own competency is something that I'm developing in it. I'm not a coder, or I'm not actually an AI expert myself, but I have been working myself, and we all are going to start seeing results that are non-binary. As a field, we have been used to seeing some binary results in terms of deciding embryo fate. We'll start seeing the change here with more complex outcomes. It won't be as black and white. There might be grays. We already explored that with Mosaicism, and everybody basically was like, oh my God, what is Mosaicism? But there's going to be more of this coming through. But there will be tools that we create that will be used to actually see the sense with the gray also. That's sort of the hope here. The paradigm change is there won't be a yes or no answer. There could be the maybe answers, and there might be places and positions where doctors and experts such as yourself will have to chat with the patient and really understand what's going on. But this will significantly improve the overall outcome of the assay and of the IVF cycle in itself. We are already looking at some data. It's very preliminary, so I did not feel the confidence to present it today. But we have looked at data with some AI models, we could eliminate 20% of embryos 100% of the times. There is no chance of those ever being implanted. And the question regarding mosaicism will at least be solved to a 20%, and then we work from there on.
It's an excellent presentation, Dr. Sagar. And it was clearly explaining the pros and cons of every technique used throughout. And the technical challenges still we are all facing, you are facing particularly. So it was nice and clear. It was not too much so that we were not going anywhere into. So kind of, it was really good. And we have got quite a few questions from Dr. Radha Pandian, I guess. Before that, I have a couple of questions for you.
Sure.
Probably some basic questions. As an embryologist, we'd all like to know. You said it was a day 5, day 6 culture, and then we are collecting the media for the sampling. So what is the volume of the culture in the, if it is a micro droplet culture, what is the total volume of the culture per drop, per embryo, and what is the volume of the sample?
So we've been working with about 15 microliters of the sample, 10 to 15. I have reduced it all the way to 5. Smaller the droplet size in culture, the better is the concentration of the DNA that is available, and that's quite expected there. We have worked with an array, from we worked with culture sizes from 15 into 30. It doesn't necessarily affect the outcome there. What we do is we have to know what is the culture size that you're working with, and appropriately we change downstream processing around it. The hope would be that by the time our chemistry, our Rosa fertility chemistry hits the market, we would want a certain kind of culture practices, and that's just something that again we've seen across the board. So
once you have taken the sample, how many ever microliters you want to have for the assays, and is the media getting refreshed again for the culture to continue until you go for a freezing or whatever?
Yes.
Or is it just
Yes. We once we have collected the media from the embryo, we leave it to the discretion of the embryologist. It doesn't actually interfere anyway with the result of the assay. So it depends. If you had a larger droplet size to begin with, then you probably are not that concerned about refreshing it. But if you have to, that's okay. And as I said, we're working both with stepwise and also continuous media. So
that's excellent. Then my other question is like the basic morphological assessment, what most of us follow here. Has there been any studies comparing the morphological assessment versus the NIPGTA results? Like what was the concurrence between them?
There are some morphokinetic studies that I have heard of, but I have not seen with NIPGTA. I don't have an idea. I apologize for that.
That's all right. There was a wonderful webinar from David Gardner during the lockdown period. He was talking about glucose uptake, and he was seriously taking us through the morphokinetics through time lapse, and he was on to AI, how he was comparing both, and then finally he went up to those uptake levels of each blastocyst versus its viability. So that's what like triggered me to have this question for you. It's not a problem at all. And Probably if Dr. Devan or me to go ahead with the questions in the chat box.
Yeah, ma'am, you can go ahead.
All right, the first question from Dr. Radha Pandian is like, can there be a problem of mosaicism here too? Does this NIPGTA in any way mimic earlier methods of proteomics, metabolomics, which are not coming up in big way now for selection of good embryos? That's the first question. Interesting.
So it's an interesting question. So would mosaicism be present here? The short answer is yes. Mosaicism, I was talking with Professor Darren Griffin, who's one of our collaborators, and we've been exploring how could we test mosaicism as a population-wide presentation. The hypothesis here being is that everybody could be mosaic, even healthy individuals could be mosaic, is something that is a possibility just naturally. I'm not saying IVF, embryos. I'm saying just naturally, is there a chance? Anecdotally speaking, I had a friend growing up, and one of her eye was green, and the other one was brown. And as she became older, around the age of 30, both her eyes turned brown. And that's sort of a good explanation, a good way of thinking that mosaicism could be there. What is unique about NIPGTA is that it won't be a sampling bias, right? You're taking DNA as it is secreted through apoptosis, necrosis, or just extracellular secretions into the media. So what you're seeing as a representation here, at the risk of sounding, it's a better representation than what you could in an embryo biopsy, because in an embryo biopsy, as I said earlier, there could be cells which are normal and abnormal sitting next to each other because of mitosis. So when you're picking it up, you're picking up, not necessarily it's an artifact. So that's sort of what it is. The question about proteomics and metabolomics is very interesting because at our core, Rosa Scientific is a proteomics platform. We are currently working on massively parallelizing protein detection. The patents that the company actually is formed around is squarely placed within proteomics. We want to work. We are also working with Dr. Munez's metabolomics assay and potentially finding a 2-point, sort of 2-point diagnostic around metabolomics and non-invasive metabolomics and non-invasive genetics. So I think the next few months, around 8 to 10 months, will have a really significant paradigm shift in the way that we are doing non-invasive assays.
Excellent. And Dr. Radha's next question is, does the extended culture to day 6 reduce the implantation rates, which would have happened by itself?
Yes, there's no denying that. But day 5, day 6, if you're comfortable with day 5, the assay works okay with day 5. We did look at some blasphemy day 3 related stuff, but the concordances were terrible. And this is when I was at Cooper. I haven't even bothered to look at day 3 after that in non-invasive. We looked at about maybe 50 odd, I think, blasphemy spent media, but it wasn't great. And the slide with the summary also has some day 3s, I think 1 or 2 day 3s, and the ploidy concordances are quite terrible. So it's a choice. I mean, whether you want to take it to day 5, explore competency that way, or yeah.
Okay. And the next one is, embryo self-correction is a known factor in nature. Are we not giving any credence to it?
It is a known factor, and as a diagnostician and as a scientist, I mean, the problem with that is that we can only relate to data that is being seen, and we are capturing our data at a certain point in time. Yeah, maybe it makes more sense, but then you know, I honestly think that giving credibility to an unknown set of data points is something that we can't hang our hat around, or we can't basically create a diagnostic around it, because whether it will self-correct or not is an if, right? I mean, if it corrects, yes, absolutely. What happens if it doesn't correct is also something that you know, and how we measure it is something that is beyond me. I have faced this question several times, and the answer is every single time that we have a set of data points in front of us with any assay, with a non-invasive or an invasive assay for that matter, and we either choose to look at it or not. I mean, that is something that we can decide, but whether the embryo will self-correct or not is an if. So
yeah, absolutely. And the next one is, does the interpretation vary with quality of embryos? Poor embryos may throw more DNA, and if they are transferred, is it good for the patient?
Quality or quantity of DNA is actually it's not that varied. We don't see huge deviation. And when you say poor embryos, I'm assuming the question is in regards to morphologically poor embryos, right? I mean, if I understand it right, it's morphologically you know, abnormal looking embryos, probably, that the question is in regards to. There's a standard deviation around which we see DNA extruded in. What we have seen, however, for example, is that with frozen embryos, freeze thawing it, you have a little bit more DNA than if you're looking at just fresh embryos. We have seen that with you know, with only changes like that, but comparing morphology, at least in the lab, we haven't necessarily seen any remarkable data point there. And thank you for your compliment, Dr. Radha Pandian, for my baby. I appreciate it. I just saw the question answer panel.
Yeah. I think that's all the questions, I guess. Thank you, Dr. Savarkar.
You're most welcome. I appreciate you having me here, and thank you for the opportunity.
All right.
Thank you.
Dr. Devan Patel, would you like to say a few words?
Yeah, it was a nice presentation, Dr. Sarthak, and I think there are a lot to think more about Mosaicism, and I was actually it was very interesting to see one of the slides, that is origin of DNA in spent media. So rather I will be having rather more questions, or maybe I would like to go through some more literature regarding what are more secretions or what are the other type of DNA which can give us a challenge or maybe which will be interesting to know when we do NIPGTA. But anyway, it was a very good presentation. Thank you. Thank you very much.
Thank you, sir. Can we hear a few words from Professor Pandian?
Sure.
Sir?
Yes, please. It is a candid presentation. Thank you very much for a nice presentation and the nice discussion you had. Can I ask you some more questions? Are you ready for it? Are you okay? Can
Yeah,
you go ahead with some more questions? Yes. Okay. What is your turnaround time?
I will try to answer them.
What's your turnaround time? I mean, when the fluid comes to you, how long do you take to give the report?
It is the same as any regular PGT assay. The amplification takes the same time, and the running of the assay also is the same time. So I think you know, the partner we are working with say 7 days.
7 days, which means you have to freeze the embryos. It's now, so it's mandatory to freeze the embryos, right?
Yes.
Okay. Now, you said DNA secreted. Is it really secreted or the DNA lost from those apoptotic cells, the necrotic cells? And where do the DNAs come from? Is it from the inner cell mass or the trophoblast?
That's a great question. That's sort of why we raised a concern around it. Is that the chance that it could be from either of these places, is possible. If you give me 1 second, let me share my screen again. So the reason I have the base pairs written around, this is not secreted DNA fragment size or necrotic DNA fragment size is something which is well, we have estimates around this. We have seen these in other biological systems. We don't know particularly as to what is the origin of cell-free DNA in the spent media at this point in time. And that is one of the endeavors that we are researching very heavily right now.
With that uncertainty, is it a big leap in faith to believe that this is going to decide the fate of the embryo? Because if you tell me that your NIPGTA tells me that this is an abnormal embryo, and I'm going to decide whether to transfer or not transfer based on that, are we making a huge judgment on an issue which is still very uncertain about the origin of the cell itself, the DNA itself? I mean, see, ultimately, as clinicians, we have to face the reality of to transfer, not to transfer.
And that is where you take the other studies that I showed into consideration here, right? Is that you have a comparison of ploidy concordance with trophectoderm as well as whole blastocyst. So if you see this, then there are groups using their particular chemistry who've looked at whole blastocyst and concordance, ploidy concordance of about 94%. Or there's also 100%, but the N is a little low on that. But the most recent one by Kuznetsov and Majumkova was 97%. Significant, a significant N there. Having said this, I agree with you. I mean, if it's a leap of faith, and that's why when with our chemistry we are exploring, we want to answer this. We are using our research dollars to make sure that we answer this question, so that clinicians and diagnosticians like you are more comfortable in stating as to the ploidy status of the embryo using the non-invasive assay.
Yeah, as you beautifully said, that all of us are mosaics. If you do enough number of cells, we are all mosaics. That being the case, are you still going to be reporting on mosaicism? Because you are analyzing, see, it causes a lot of confusion for the patient and to the clinician. You report embryo as mosaicism, and we are confused whether to transfer or not to transfer. This is number one. Number two, at the moment, what is the positive predictive value and what is the negative predictive value?
We are showing upwards of 99% PPV. To answer your first question, though, to answer your first question is that whether we'll report on mosaics or not. What we want to do at Rosa is we are using huge data sets, Dr. Pandyan, to basically weed out the true untransferable mosaics. And we already have prelim data. The thing is that our scientists write down some program, and then there's a huge amount of validation that will go before we come out and offer it as a product. But we are saying that just looking, brute forcing your way through a huge amount of data and actually running it through our models, you might be able to actually distinguish between the mosaics that can implant and that won't implant. So that is a deep learning project that is on our platter. So we might actually report mosaics only when we are absolutely certain that they will not implant. And we'll present result for it. The rest of them, will be your choice. Will be totally your choice in terms of how you want to proceed with it.
Okay. Thank you very much.
You're welcome.
Thank you very much, Dr. Sarthak. It was a wonderful presentation. We have come to the end of the meeting. I would like to thank our speaker for the day, Dr. Sarthak Savarkar, our chairpersons, Ms. Sarvana Devi Shivanandam and Dr. Devan Patel, for this wonderful session and discussions. And I would also like to thank our Professor Pandian and Dr. Radha Pandian for the discussions again. And I would also like to thank the Sun Pharma Senior Division, their GM, Ms. Rashmi Parekh, Mr. Sapan Shah, and Mr. Balaji and Ranjit, and the entire technical support team. And thank you, participants, for joining this meeting. We shall meet you all in the next IFRIF meeting next month. Thank you once again.
Thank you,
Thank you all. All the best to you for your PhD and for other things to come. Good day.
Vanakkam.
Good day.
Bye.
Vanakkam. Good day. Namaste to all of you.